Very good evening. I hope so. My screen is visible. Am I audible? Guys, am I audible? Is my screen visible? Can someone put in the chat box so that I can just continue? Am I audible? Am I clear? With the hope that I'm audible and I'm clear, I'm just starting the session. First, in the beginning itself, I'm just telling that please be interactive in the session so that it will be very interesting. So biochemistry, you have on 9th and 10th of November, you have your exam for biochemistry. And I hope so you have at least gone through once all your chapters while you're reading, isn't it? So it's very easy. Basically, the biochemistry subject is very easy. So I don't want you to just be tensed. So we will try to cover as much as possible. By conceptual, we will be covering. So everything is from your textbook only. So please listen and take notes so that it will be benefit for you. So without further delay, let me begin with today's topic, that is going to be a molecular biology part 1. So today we will be dealing with the basic chemistry of all your genetic material. So your DNA, RNA, and what are the nucleotides that are present. That is exactly what we are going to deal with today, the chemistry of nucleic acid. So once you understand the chemistry of nucleic acid, of course, it will be very easy for you to understand the transcription, translation, replication, post-transitional modification, everything. Believe me, guys, everything is very easy once you understand the nucleotide and its concept. So that's what I'm just going to try today to make you understand about it. So what is nucleic acid basically? Nucleic acid is nothing but they are the polymers of purine and pyrimidine. What is this purine and pyrimidine? All these are Greek and Latin word, isn't it? So I'll tell you, first let's see the nomenclature. How is these structures are named? How is these structures are named? That is what we are going to see first. Purines, the word purines, everything is going to be purine, and we are going to have one more structure called as pyrimidine. Okay, purine mudira ellame, it has SINE. So it is going to have SINE at the end. What are the two SINE in end? Are two things which we know. One is going to be your adenine. Okay, your adenine, and you know guanine. Adenine, and other one is your guanine. These two are your purines. Okay, when this adenine combines with your ribose, you call it as adenosine. Okay, you call it as adenosine. Whereas when the guanine, it combines with your ribose, you call it as ribosine. Do we call it as ribosine? No, we call it as guanosine. So you have two things, adenosine, guanosine. Both these things are called as purine. As I told you, INE, it ends with INE, right? So all your adenosine and your guanosine, everything ends with INE. So these two are going to be your purines. Now, next one is going to be your pyrimidine. So what is your pyrimidine? Pyrimidine is nothing but everything that is going to end with DINE. So both the pyrimidines which we are going to discuss, they are going to be ending with DINE, pyrimidines. So how we are going to write? So there are two important things. One is your cytosine and other is going to be your uracil. So when both of them going to combine with ribose sugar, they give you cytodine. I told you, right? It ends with DINE, and you have this uridine. URIDINE. So these are the structures that are going to be making up your DNA, making up your RNA. But you have one important difference. Do you understand that difference? Which is the one that is present in the RNA and which is the one that is present exclusively in DNA. So I told you cytosine, I told you uracil. There is a third one called as thiamine. So this thiamine structure, when combines with your ribose, it forms thymidine. So this thymidine is exclusively available in your DNA and uridine is exclusively available in your RNA. So this is what I want you all to understand first. So is it very easy, right? The nomenclatures of this purines, pyrimidines. So this is the first thing what I want you all to understand. So base is going to be a purine pyrimidine. This purine and pyrimidine combines with your sugar, and along with sugar, it also takes one phosphate molecule to form the nucleotide or to form the nucleotide. Let's see one by one. So before moving into those concepts, you know that the basic genetic material of a human being, of an animal, of everyone is going to be your DNA. Okay, this DNA, if you want to replicate the DNA, see, I am a person. Now I want a clone of myself. If I want a clone of myself, my DNA has to be replicated. But the problem is the DNA cannot be directly replicated into a DNA. The DNA has to replicate first into RNA. After that, that mRNA, or we call it as a messenger RNA, and they will be like giving us a base to convert and form a new DNA. Similarly, the DNA is not just needed for the replication process. The DNA is not just needed for the cloning of the cells, but it is also needed for the production of enzymes. You need enzymes, you need proteins in your body. So these proteins are also synthesized from your DNA. That is the why you call this DNA as the basic functional genetic material of our body. They carry the codons for production of the proteins in our body, for production of enzyme in our body. So that is what we are going to deal it in this entire molecular biology. We are going to see how this DNA is synthesized. The synthesis of DNA, we call it as, so this DNA synthesis, we call it as a replication. So that is the first process that is going to occur. Then this DNA synthesis, once it occurs, it has to replicate. So it has to form a RNA. So how it forms RNA? This process is called as transcription. This process is called as transcription. So first one is your DNA synthesis, that is called as replication. Then you have the transcription, by which you get a RNA, and from this RNA you will be getting a protein, isn't it? So that protein formation is called as your translation. That protein formation is called as a translation. Okay, now I told you that all your DNA and your RNA is going to be made up of nucleotide. So this is called, it's made up of nucleotide. They are going to be made up of nucleotide. Okay, this nucleotide is actually made up of two components. One is going to be your nucleoside and other is going to be your phosphate. Okay, one is a nucleoside and other is a phosphate. This nucleoside, again, it is going to be combined of a sugar and a nitrogenous base, and this nitrogenous base is what we call it as purine and pyrimidine. So one of, in the beginning I told you nomenclature, isn't it? So that nomenclature, we had a purine and a pyrimidine. This purine and pyrimidine, what happens is they combine with a phosphate molecule to form a nitrogenous base. This nitrogenous base, again, it combines with a sugar. So you want a sugar, isn't it? So what type of sugar do you use here? Why you use a ribose sugar instead of deoxyribose sugar? All these questions you will be having in your mind while you are reading your molecular biology. And today the answers for those questions are what we are going to be derived at the end of the lecture. So this is a pyrimidine base. You have a three structures: cytosine, uracil, and thymine. And this cytosine, uracil, thymine are the pyrimidines. Okay, I told you what is a nucleoside. A nucleoside is a nitrogenous base along with a sugar. So what type of sugar you use? That was my question. You use a ribose sugar. Why don't you use a deoxyribose sugar? What is the answer? You need a stability for your nitrogenous base, isn't it, guys? For having the stability of your DNA RNA, you need ribose sugar and you can't use a deoxyribose sugar. Now you understood, right? Why do you use ribose instead of deoxyribose? We use deoxyribose. Okay, we use deoxyribose. Okay, and why not ribose alone? Okay, that was the question, isn't it? So we use deoxyribose because the hydroxyl group at the 2 position can participate in a reaction that cleaves your phosphodiesterase bond. So in the hydroxyl group, the DNA is absent. அப்ப DNA absent இருந்ததுன்னா polymer எப்படி இருக்கும்னா ரொம்ப ஸ்டேபிளா இருக்கும். Thus DNA can be acting a very stable. So RNA is degraded within 30 minutes, whereas DNA lasts for lifetime. Why? Because ribose is present in your RNA and deoxyribose is present in DNA. Now you understood, right? So we in a human being, we have deoxyribose sugar, whereas in a animal or in a plant, you are more than animal, I can tell a plant, because the plant and prokaryotes will be usually having this RNA material. This RNA material will be having a ribose sugar and not deoxyribose sugar. Now you can understand that the deoxyribose gives you a stability, whereas ribose sugar will be easily cleaved by 30 minutes. Therefore, deoxyribose is used in the DNA production. Is it clear, guys? Now, after understanding this, so what I wanted to add up on here is you have two types of sugar. One is your deoxyribose sugar and other is going to be your just a ribose sugar. So this just a ribose sugar is used in RNA, whereas deoxyribose is present in the DNA. So with this, we are completing the basic chemistry. So I will show you the structure. Don't worry. So I told you, right? Adenine plus ribose gives you adenosine. Guanine plus ribose gives you guanosine. Uracil plus ribose gives you uridine. And cytosine plus ribose gives you cytidine. How to remember that? I told you. Pyridines, pyrimidines, and purines. Purines will be ending with INE. Therefore, your adjacent structure, adjacent material will also have a INE at the end. Whereas pyrimidine ends with DINE. Therefore, your uridine and your cytidine are going to end with DINE. There's another one purine called as hypoxanthine. There is another purine called as hypoxanthine. What is this hypoxanthine? This hypoxanthine combines with your ribose to form inosine. You would have already seen in your physiology, you would have told inosine triphosphate, IP3DAG mechanism. In all these things, you have this inosine. How this inosine is produced? Inosine is produced by hypoxanthine combining with ribose. Similarly, xanthine, when combines with ribose, they form xanthosine. So these two are exclusively present for the production of enzymes. These two are exclusively present for production of enzymes. Then coming back to the deoxynucleoside, okay, or we can call it as DNA, you have only 4 components. So one is your adenine, guanine, cytosine, and thymine. They form deoxyribose adenosine, deoxyribose guanosine, deoxyribose cytidine, deoxyribose thymidine. Okay, now tell me what is a nucleotide. I already told you the answer for this question. Nucleotide are nothing but the nitrogenous base when combines with the sugar, when combines with the phosphate. So all these 3 molecules are going to be together. We call it as a nucleotide. And these are the ones that are going to be making up your DNA and your RNA. Okay, so this is how your nucleotide will be present. So you have a nucleotide. So this is your base pair. This is going to be your base pair. Then you're going to have your sugar content and you will be having phosphate. This phosphate, what happens is they are going to be having a connection with the adjacent nucleotide. So what happens when there is a single phosphate in a nucleotide, it is called as mononucleotide. When they are going to add 2 or 3 phosphate, they trade into di or triphosphate. When you're going to add more and more phosphate molecule, what happens is they are going to be combining with another nucleotide. Thus, it forms a chain of nucleotide, and this chain of nucleotide, you call it as a, what do you call a chain of nucleotide? A chain of nucleotide is only called as your DNA or RNA. When you're going to just, when you're going to see, we have a chromosomes, isn't it? When you open the chromosomes, you're going to have a DNA. When you're going to open that and when you're going to extend and completely make it like a chain, it extends from Earth to Moon. அந்த அளவுக்கு பெருசு நம்மளோட DNA materials. அப்ப இது எல்லாமே என்ன ஆகுதுன்னா, everything is packed into a small molecules. How this packing occurs, guys? And this packing is only the end product of your transcription and translation and post-transcriptional modification and everything. Okay, that is what we are going to discuss today. And till now, is it clear, guys? Can someone put in the chat box? Till now, is the chemistry clear? Once the chemistry is clear, then only we can move into the adjacent part. Is it clear, guys? Is the chemistry clear? Can I move into the adjacent one? Yes. The next topic is going to be the properties of your nucleotide. So this can be asked as a 2-mark question for you guys. So how is the nucleotides present? This nucleotides, actually, they will be having a partial double bond character. So I will just give you a highlighting. I'll just highlight it. Please make sure that these points are never forgot in your life. So partial double bond character. So it will be having a double bond, all your nucleotide, and your nucleic acid will be absorbing UV light. Okay, that's all. No need of this one. The nucleic acid absorbs UV light. That's all. And then you have your purine, pyrimidine. All these purine and pyrimidine are going to be hydrophobic in nature. They are going to be hydrophobic in nature, and they form a lactoform predominant. So they form lactam group of molecule at pH of 7 at a neutral characteristic. So then what you have to understand is you have to understand certain important molecules. Okay, what are those important molecules? Number one, ATP. I think everyone know ATP is a basic energy of your body, universal energy currency. You would have learned in your 12th standard, 11th standard and all. So they are going to give you the energy for all the processes which are going to occur in the coming all the processes. Okay, so ATP is going to give you the energy. And you have NAD, NADP, FAD, coenzyme A. They are going to be a enzyme cofactors. So it is an enzyme cofactor. And you have the cyclic AMP and cyclic GMP. They are acting as a secondary messenger in all your process. So now slowly we are going to move into your molecular biology, guys. We are going to move one by one. We are going to move into your molecular biology, crux of molecular biology. And first, for that, you need to understand the basics. We have understood what is a purine, what is a pyrimidine. Next, we have understood what are the important part which are going to take part in your process: ATP, NAD, and your cyclic AMP, cyclic GMP. Then we have this S-adenosyl methionine. So this S-adenosyl methionine, they are going to be helping you for giving you the intermediate products. So you need a product, you need a raw material, isn't it? So this 5-adenosyl methionine is going to give you a raw product, and 3-phosphoadenosine 5-phosphosulfate. They are going to be having a sulfate group, and it is a most common coenzyme in your sulfotransferase enzyme, sulfotransferase reaction. So that is what we are going to go into it slowly. Now, I am just skipping all these things. One thing you need to understand is your first thing, DNA. What is a DNA? You need to understand what are going to be the different parts of the DNA. You have your time. What are the different things you have in your DNA? You are going to have cytosine, you are going to have thymine, you are going to have adenine, you are going to have guanine. These are the 4 things that make up your DNA. And this is going to be the genetic material of our body. They are going to give the genetic code for production of protein, for production of enzymes. And that is why they are called as master of cells. So we call them as master of cells. Okay. Now, this is the structure of DNA. So your DNA is actually like a helical in structure. Okay, well, it is a helical in structure. I'll be discussing in detail later also. For now, you remember that you have a DNA helical in structure. You have different structures of DNA. Okay, so these DNAs are arranged in the hydrogen bond, which I will be discussing later. For now, you remember DNA, that's all. Then you have another structure that is called as RNA. Since you already saw DNA is a double-stranded helical structure, whereas RNA is a single-stranded helical structure. That, okay, you have one rule called a Chargaff rule. I will be discussing again. This Chargaff rule is applicable only for your DNA. It is not applicable for your RNA. What are those Chargaff rules? I will discuss it later. Okay, I'll discuss it one by one. For now, what you're going to remember, already you know what is purine, what is pyrimidine. You know how your purine and pyrimidine combine to form a nucleotide. And you also know this nucleotide are going to make up DNA and RNA. So you have a DNA which is a double-stranded helical structure. You have RNA, which is a single-stranded helical structure, and your DNA is going to be making up a genetic material, whereas RNA is going to make up proteins in your body. ஒண்ணுமே இல்ல. ஒருத்தர் வந்து ஜெனடிக் மெட்டீரியல் கொடுக்குறாங்க, இன்னொருத்தர் புரோட்டீன் சிந்தசிஸ் ஹெல்ப் பண்றாங்க. So DNA-வும் required, RNA-வும் required in our body. So main component of our genetic material is our DNA, but RNA is also present in our body. That is what I want you to remember. Usually in our early childhood, what we would have remembered is DNA present in the eukaryotes, RNA present in the prokaryotes. It's not like that. The DNA is a building block of the humans, And RNA is the building blocks of your prokaryotes. But at the same time, the RNA is also present in our humans. That is what I want you all to remember. Now, you have different types of RNA basically. One thing which we will be which we have interest is going to be your messenger RNA and ribosomal RNA. These are the two RNAs of interest for us because they are going to be participating in the humans, and that is why we will be having a more interest on reading them. Okay, skipping all these things. Next, I am just moving on towards the very important topic, that is going to be your purine metabolism. Now you know that how your purines are going to be. Okay, do you have any doubt till this? Shall I move to the next one? You know that your purine is going to be a very important thing. So before starting, I just want to give you a certain terminologies which you need to understand. What is N1 called as? N1. உங்க bookல காமனா இந்த மாதிரி கொடுத்திருப்பாங்க. Almost you will all will be confused. What is N1? N1 is your aspartate. N1 is your aspartate. C2C8 இது என்னது? They are going to be nothing but formate. Okay, C2 and C8 are going to be formate. And you have N3 and N9. N3, N9 இவங்க பேருமே வந்து they are going to be glutamine. Okay வா? And C4, C5 and N7, they are going to be glycines. Okay, and you have the C6 will be comprising of CO2. What is this? What is this I am writing? Am I stupid? No. I'll just give you how this is going to be. I'm just going to represent everything as a structure. Once you understand these 5 things, you can easily represent the structure in your exam. See, what I told you, N1 is an aspartate. So first one, I'm just putting N1, and this N1 is nothing but your aspartate. Okay, well, this N1 is your aspartate. Now, after N1, what happens is, so you're going to have your second chain, second part. That is going to be C. C2 is going to be there. C2 is going to be there. அடுத்தது என்னது N3. See, third position la N இருக்கு. So I am just putting N3. Then C4 is there. Of course, yes, C4. Then C5 is also there. Then you're going to have a C6. Okay, wow. So now you understood. N1 is your aspartate. C2 is going to be formate. Okay, C3 கிடையாது. N3, we have N3. 3 position la we have glutamine. In the 3 position, what we are having, in the 3rd position, we are going to have glutamate, and 4th position, we are going to have glycine. Can you all write it along with me, guys? That will be helpful to you. C2 is going to be your formate, and N3 is going to be again glutamine. Okay, glutamine, and you're going to have C4. C4 is your glycine, and you're going to have your C5. C5 is again glycine, and you're going to have your C5 is again going to be glycine. And what is your C5 is going to be glycine, and this is also glycine. I just represent as G. And your C6 is going to be carbon dioxide. Now you have C4 and C5, isn't it? So at that position, we are going to form a new ring. Okay, so this is going to be C4. So we ended at C6, isn't it? So we ended at C6, so we form it at C7. So இப்படியே நம்ம form பண்றோம். C6 on the side போகும், C7 in the side. So C7, then you're going to have your C8, then you're going to have your ammonia, NH. Okay, then you're going to have your nitrogen again here. So nitrogen attached to your NH. Okay, well, nitrogen attached to your NH. Now tell me N9. Where is your N9? N9 is again going to be your glutamine. N9 is again going to be a glutamine. C7 is going to be your, what is C7? Is it C7 or N7? It's N7. N7 is going to be your glycine, and C8 is going to be your formate. Now is it clear? One, in the structure நீங்க மக படிக்கணும்னா it will be very difficult for you. But instead of mugging up the structure, what I am telling is mug up this one. Memorize this one. ஓகேவா? இது வந்து பயோகெமிஸ்ட்ரியில நீங்க ஃபர்ஸ்ட் இயருக்கு அப்புறம் இது எதையுமே நீங்க படிக்க போறது கிடையாது. இந்த ஸ்ட்ரக்சர்ஸ் எல்லாம் யூஸ்லெஸ். பட் சின்ஸ் யூ வாண்டட் டு ரைட் இன் யுவர் எக்ஸாம், for that, what I am telling is mug up these 5 points. Mug up these 5 things. What are the 5 things which you are going to memorize? N1 aspartate. Then you have your C2C8 formate. So I can remember C2C8 formate. Then you have N3, N9. So எப்பயுமே N எதுவா இருக்கும்னா all your amino acids. Aspartate is an amino acid. So that is N1. You are going to have your glutamine. Glutamine is an amino acid. So you're going to have your N3 and N9. Then you have your glycine formate. All these are going to be your alcohol, carbon dioxide. You need C. Okay. So that for formate is C2C8. and glycine is going to be C4, C5 and N7. So glycine is again, it is going to be a combination of, it's going to have carbohydrate component in the nitrogen. So it's a carboxy amino acid. Therefore, you have the C4, C5 and N7 that is going to give you glycine, and you have the C6 component that is going to be carbon dioxide. Can you represent this in your exam, guys? Now, I hope that you can represent with these 5 points, you can represent the purine structure. Once you understand the purine structure, then everything becomes easy. Once you understand the purine structure, everything becomes easy. What is that everything? I'll tell you. Now, you are going to talk about the purine. Okay. So what happens is, you are going to take a nucleic acid. So you're going to take a nucleic acid. So you have this ingested nucleic acid, ribonuclease, deoxyribonuclease, everything. So you have your damaged DNA. All these damaged DNA and damaged RNA are going to be broken down into nucleoside. Okay, nucleotide. They are going to be broken down into nucleoside, nucleotide, and finally they will be broken down into purine and pyrimidines. So they will be breaking down into nucleic acid. So all your dietary things, whatever you are eating, they are also going to have RNA. They are also going to have a nucleic acid. They are going to be broken down to form purines and pyrimidines. Once they are broken down to form purines and pyrimidines, what happens? With this purines and pyrimidines, the de novo nucleotides are going to be synthesized. What is the meaning? Your body inherently is going to produce your DNA and RNA out of it. So how do they produce DNA and RNA out of it? So they are going to produce DNA and RNA by the production of purines and pyrimidines. Once purines and pyrimidines are synthesized, once you synthesize the purines and pyrimidines, it will be easy for you, isn't it? So that is called as a de novo synthesis of purine. So this purine ring is assembled on the ribose 5-phosphate. You have this IMP, inositol monophosphate, which is going to be the parent nucleotide, from which you are going to get this AMP, GMP. I told you, right? All this AMP, GMP, inositol, all are going to be acting as a cofactor in your DNA production. Okay, now this is going to be the structure. I already told you this is going to be the structure of your purine ring. Now you understood, right? This is a structure of purine ring. What happens is, you have ribose 5-phosphate. Ribose 5-phosphate is nothing but the sugar moiety. This sugar moiety in the presence of PRPP synthetase. So this is what will be given in your textbooks and all. Now you understood purine, the end product oda structure namakku theriyum. In the end product structure eppadi varudhu abingadha nama paaka porom. So first you are going to have your ribose 5-phosphate. Ribose is a sugar moiety. That sugar moiety is present. Now on this sugar moiety, when there is action of PRPP synthetase, PRPP synthetase enna pannuna, ATP AMP convert pannum. Appa ATP is converted into AMP, then what will be the end product? So you will have a ribose 5-phosphate. Along with it, you have 2 more phosphates with you, isn't it? So you have your ribose 5-phosphate. Along with it, you have 2 more phosphates with you, because your ATP has converted into AMP, releasing 2 phosphate. This 2 phosphate combines with your ribose 5-phosphate to form what? What are they going to form? Someone, can you put in the chat box? I hope I am making it clear. What happens is, you have a ribose 5-phosphate. With this ribose 5-phosphate, 2 more phosphates are going to come and add. When 2 more phosphates come and add, you call it as phosphoribosyl pyrophosphate. You call it as phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. Pyrophosphate. So enna agudhu ingena onnume illa, romba simple ah you have this ribose 5-phosphate. Along with this ribose 5-phosphate, what happens? You are going to add, what you are going to add? 2 phosphates along with it to form this phosphoribosyl pyrophosphate. So first is the clear. Idla structures la marandringa. Just steps na vechkonga, and the steps can be asked in your exam. Okay, so you are going to have your ribose 5-phosphate. When this ribose 5-phosphate gets phosphorylated in the presence of ATP, in the presence of pyrophosphate synthase. So you are going to have your phosphoribosyl pyrophosphate, PRPP synthetase. So you are going to have PRPP synthetase, isn't it? So on the PRPP synthetase is going to give you phosphoribosyl pyrophosphate. It is going to give rise to phosphoribosyl pyrophosphate. Now you understood, right? Now what you have in your hand? You have phosphoribosyl pyrophosphate. Now this phosphoribosyl pyrophosphate, it is going to be, there is going to be addition of N9, nitrogen. So there is going to be addition of nitrogen at 9th position. So how you are going to add nitrogen at the 9th position? So in the presence of PRPP, glutamyl aminotransferase. So there is meaning. You have your PRPP. The PRPP, glutamyl aminotransferase. You are going to give a phosphate. You are going to give a phosphate. Along with it, you are going to give a glutamyl. So you are going to give an amino acid. You are going to give a nitrogen for it. So PRPP, in the presence of PRPP, glutamyl aminotransferase, gets converted into phosphoribosyl amine. ஏனா நீங்க என்ன பண்றீங்க amine ஒண்ண ட்ரான்ஸ்ஃபர் பண்றீங்க. So nameலயே இருக்கு பாருங்க, PRPP glutamyl amidotransferase. You're going to transfer amine. So what happens? Your phosphoribosyl pyrophosphate is going to get converted into phosphoribosyl amine. Pyrophosphate அப்படின்னு சொல்லாம இப்ப அமைன் வந்துட்டதுனால we call it as amine. So phosphoribosyl pyrophosphate பதிலா it gets converted into a phosphoribosyl amine. Now you understood, right? So now you have 5 phosphoribosyl amine. So it started from your ribose 5-phosphate. Ribose 5-phosphate get converted into a phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate, after adding the amino group to it, you call it as 5-phosphoribosyl amine. இப்ப இதுக்கு நடுவுல the 5-phosphoribosyl amine வந்து இதுக்கு நடுவுல உங்களுக்கு 9 steps இருக்கு. But unfortunately or fortunately you don't need to read that. Please leave that. இத மேல படிச்சீங்கன்னா கன்ஃபியூஸ் ஆகும். So first step PRPP synthesis. Second step is going to be your addition of N9 group. That is a rate limiting step. So this is what you need to remember. Okay, so this step is what important you need to remember. அதுக்கப்புறம் ஒரு 9 steps இருக்கு. அந்த 9 steps I am just skipping. So I am just skipping it. And now what I have is, after the ring closure, after the ring closure, what I am going to have in my hand is going to be, your inosine monophosphate. So after completion of your 5-phosphoribosyl amine, அதுக்கப்புறம் ஒரு 9 steps இருக்கும். தயவு செஞ்சு அந்த 9 steps skip பண்ணுங்க. It has no sense at all to read that. So அந்த 9 steps அப்புறம் என்ன ஆகும்னா, it forms inosine monophosphate. So how it started? Ribose 5-phosphate. After that, it gets converted to a phosphoribosyl pyrophosphate. It gets converted to 5-phosphoribosyl amine. In the 5-phosphoribosyl amine, after a 9 steps, it comes into form a inosine monophosphate. Now you have a inosine monophosphate in your hand. This inosine monophosphate, in the presence of inosine monophosphate dehydrogenase. So dehydrogenase, what it is going to do? It is going to remove a hydrogen component from your IMP, inositol monophosphate. So who has to remove the hydrogen? So someone has to come forward to take hydrogen from your molecule, isn't it? So அதுதான் வந்து NAD+. So what your NAD+ is going to do is, he is going to come and remove a hydrogen from you. So it is going to remove hydrogen from you and they are going to form NADH plus H plus. Okay, so ultimately what we got is, when inosine monophosphate, when you're going to remove hydrogen component from it, what you get is xanthosine monophosphate. இதுல ஸ்ட்ரக்சரை மனப்பாடம் பண்ணாதீங்க, ஸ்ட்ரக்சர் தேவையில்லை, பியூரினோட ஸ்ட்ரக்சர் மட்டும் தெரிஞ்சா போதும். What you want to remember is, it started as a ribose 5-phosphate, it went as PRPP, it went as 5-phosphoramine, then you got IMP after 9 steps. After 9 steps, you got IMP. This IMP is going to form xanthosine monophosphate, and this xanthosine monophosphate, in the presence of transaminidase, gets converted into a guanosine monophosphate. Now you got your primary base for your nucleotide, isn't it? Guanosine monophosphate. Similarly, the same IMP monophosphate can also get converted into adenosine monophosphate. So it can get converted into adenosine monophosphate. It can get converted into a guanosine monophosphate. So now you have two important things: adenosine monophosphate, guanosine monophosphate. So what is important here is, you have certain regulations. Okay, you have certain regulation. The first one is the concentration of PRPP. So the amount of PRPP formed from your ribose 5-phosphate, that is going to decide the amount of your guanosine monophosphate or adenosine monophosphate generated. So this is the first point you need to understand. So the rate of PRPP synthesis depends upon the ribosyl phosphate. So ribose 5-phosphate and the activity of your PRPP synthetase enzyme, isn't it? நான் அதனாலதான் உங்களுக்கு இம்பார்ட்டன்டான ஸ்டெப் மட்டும் தான் சொன்னேன். இந்த ஃபர்ஸ்ட் ஸ்டெப் இஸ் வெரி இம்பார்ட்டன்ட். ஓகேவா? சோ அந்த ஃபர்ஸ்ட் ஸ்டெப்ல நீங்க இது எதுக்கு நான் படிக்கிறோம் எல்லாம்? Can someone just realize why should we read all these things? So ஒரு DNA synthesis கேன்சர்ல ஒரு DNA எக்ஸஸா ப்ரொடியூஸ் ஆகுது. சோ ஒரு புரோட்டீன் நிறைய ப்ரொடியூஸ் ஆகுது. சோ அதனாலதான் கேன்சர் வருது. சோ அப்ப அந்த கேன்சரை நம்ம இனிபிட் பண்ணணும்னா நம்ம என்ன பண்ணணும்? அதோட ஸ்டெப்ஸ் ப்ரொடக்ஷன் ஆஃப் டிஎன்ஏவை நம்ம இனிபிட் பண்ணணும். அப்ப ப்ரொடக்ஷன் ஆஃப் டிஎன்ஏவை இனிபிட் பண்ணணும்னா ப்ரொடக்ஷன் ஆஃப் த பேஸ் பேர்ஸை இனிபிட் பண்ணணும். அப்ப ப்ரொடக்ஷன் ஆஃப் பேஸ் பேர்ஸை நம்ம இனிபிட் பண்ணணும்னா we have to inhibit the production of purines and pyrimidines, guys. So you have to inhibit the purine and pyrimidine. You need to understand at what particular area you need to inhibit the process so that it will be helpful for you for deriving a new drug. Okay, so and the new drug da purines inhibitor. So purine inhibitor, purine nucleotide inhibitor எங்க ஆக்ட் ஆகணும்? PRPP synthetase அப்படிங்கிற என்சைம்ல ஆக்ட் ஆகணும். Similarly, your adenosine monophosphate, guanosine monophosphate as a feedback mechanism. So நிறைய AMP and GMP you got now, it will feedback inhibit the production of your AMP and GMP. Okay, so this is the entire step. So I want you to remember only this. ஓகேவா? இதுல இதுக்கு மேல நீங்க அந்த 5-9 ஸ்டெப்பையும் நான் வச்சுக்கணும்னு நினைச்சீங்கன்னா இட்ஸ் யோர் விஷ். I again tell you that you can remember if you are very much capable of it, but at the same time if you want to pass your exam, if you wanted to even get a seat in the NEET PG exam, even if you wanted to get a good rank in your NEET PG, you need to remember only this much. You don't need to remember the 9 steps. Is it clear? Bookla ipi edhikonga the 9 steps thevai illa. So ribose 5-phosphate in the presence of ATP. Once again I'll just revise it. You have your ribose 5-phosphate in the presence of ATP, in the presence of PRPP synthetase, it gets converted into PRPP. This PRPP in the presence of glutamine, in the presence of glutamine, in the presence of aminotransferase. So you have PRPP, glutamyl aminotransferase, which will be giving you 5-phosphoribosylamine. This 5-phosphoribosylamine after 9 steps, it comes into formation of IMP, inositol monophosphate. After the inositol monophosphate formation, they are going to be acted by the GDP. Okay, they are going to be acted by the GTP. GTP converts into GDP to give you a phosphate. This phosphate combines with your inositol monophosphate to form a adenosine monophosphate or guanosine monophosphate. They are going to form you the basic of your energy cell, that is your ATP. இப்ப புரியுதா? இந்த ATPங்கிற basic energy cell இந்த processல இருந்தா வருது. So all your energy mechanisms, they require the production of your ATP, they require the production of your purines. That much important is this step. Okay, is it clear? So what you need to understand here is you need to understand what is a rate limiting enzyme. What is the meaning of rate limiting enzyme? I told you rate limiting enzyme is going to be your phosphoribosyl pyrophosphate glutamyl aminotransferase. So this is going, this step, second step we called it as a rate limiting enzyme. Rate limiting enzyme. Okay, what is rate limiting enzyme? Rate limiting enzyme is nothing but the longest time taken for the process. Okay, the longest time taken for the process and the particular process and help பண்ற enzyme is called as your rate limiting enzyme. Okay, உங்க rate of production limit பண்ணுது because நிறைய நேரம் அந்த process நடக்கிறதுனால rate of steps are going to be limited. That's why you call it as a rate limiting step. Okay, then you have your regulatory enzyme. Who is a regulatory enzyme here? PRPP synthetase. PRPP synthetase is a regulatory enzyme. I hope so you are clear with this. Okay, next one is going to be de novo synthesis. De novo and raw material and purine synthesis பண்ணதில்ல, அதுதான் de novo material, de novo synthesis. Okay, that is what they gave in your textbook. What is a salvage pathway? Remember, your brain is going to be having a low level of PRPP aminotransferase. PRPP aminotransferase brain la illa. Okay, so appa brain la illana your erythrocytes and polymorphonuclear leukocytes, they cannot synthesize 5-phosphoribosylamine. Okay, 5-phosphoribosylamine synthesis ஆகாது. அப்ப என்ன ஆகும் அப்படின்னு பாத்தீங்கன்னா, the body has a wonderful mechanism that inside your brain, it is going to act as a salvage pathway. So what happens is your adenine, guanine are broken down to form AMP and GMP. Your hypoxanthine will be breaking down to form IMP. That means all the breakdown products are reutilized inside your brain itself, so that it does not depend on outside for the purine on pyrimidines to come up. Okay, wow. So that's very important. Can you see here? This adenine breaks down to form AMP. Ipoxanthine breaks up to form IMP. Guanine breaks up to form GMP. Ultimately, what we need this IMP, inositol monophosphate. This inositol monophosphate we are deriving from the ipoxanthine. So your PRPP dependent phosphoribosylation of purine. Okay, well, this is your salvage pathway. You need to understand this salvage pathway. Now, what you need to understand is you need to understand the inhibitors of this purine synthesis. So that we call it as a purine analog. Purine analog na purine madriye irukra onna na kudutena, appa nama body gets confused. They think that purine is excess and they stop the production. Then competitive inhibitors. Sometimes what happens? Purine போய் utilize ஆக வேண்டிய இடத்துல இவங்க போய் utilize ஆயிடுறாங்க. So that உங்களுக்கு automatically what happens, you get what you required. So your body will tell, okay, I got what I required, so stop the production. Then arrest your cell division. You can arrest the cell division directly so that purine metabolism does not occur. Then this is a point where you need to understand these processes are the one which is used in the anti-cancer drug. I told you, right? Anti-cancer drug like your mercaptopurine, cytosine, you have your azathioprine. இதெல்லாமே next year படிப்பீங்க. So mercaptopurine, cytosine arabinoside and your azathioprine. இவங்க மூணு பேருமே they are going to act by inhibiting the purine synthesis. They are going to inhibit your purine synthesis. Okay, va? Then once you got your purines, now what happens is you need to catabolize it, isn't it? So you need to degrade it. You need to send it out, isn't it? So and the sending out is called as your catabolism of purine. That sending out is called as catabolism of purine. Because of this catabolism, because of the improper catabolism, you will be getting certain diseases in your body. Okay, that's the only purpose of what we are going to read, this catabolism of purine. So you have this purine has to get converted into uric acid. This purine has to get converted into uric acid. That is called as the formation of uric acid. How is your uric acid formed? Remember, guys, you have this adenosine. This adenosine, first it gets converted to iosine. So in the presence of adenosine deaminase, this is the first step. I want you to note it down. Adenosine deaminase is required for breakdown of adenosine to form inosine. Okay, wow. Adenosine, ionosine, matra yaru, your adenosine deaminase. Once this inosine is formed, in the presence of purine nucleoside pyrophosphorylase, in the presence of pyrophosphorylates, what happens? They get converted into ipoxanthine. After ipoxanthine gets converted into xanthine, xanthine gets converted into uric acid. Similarly, you have your guanosine, guanine, xanthine, uric acid. So ultimately, xanthine converted again, the xanthine gets converted into uric acid. This is the entire process, guys. Can you understand this? Can you understand till now? So you have adenosine, you have guanosine. They are going to get broken down in the presence of adenosine deaminase. You have your adenosine deaminase to form inosine, and inosine in the presence of purine nucleoside pyrophosphorylates, it gets converted into ipoxanthine. Similarly, guanosine in the presence of purine nucleoside phosphorylates gets converted into guanine and then xanthine. So ultimately, this xanthine is going to get converted into uric acid. So this uric acid, in the presence of uricase, gets converted to allantoin. In the presence of allantoinase, it gets converted to allantoic acid. In the presence of allantoinase, it gets converted to urea. This urea, in the presence of urease, it gets converted into ammonia. So எப்படி ஒரு process வருது பாருங்க. யூரிக் ஆசிடாவே டைரக்டா எக்ஸ்க்ரீட் பண்ணலாம்ல. யூரிக் ஆசிடாவே டைரக்டா எக்ஸ்க்ரீட் பண்றது யாரு? காக்கா, பறவைகள், all your pigeons, everything they excrete it as a urea itself. They excrete as a uric acid itself. So in the form of allantoin, it is a water soluble. But what happens is, in humans, when I'm going to consider humans, it is not going to be directly excreted as uric acid or urea. It is excreted as ammonia. It is going to be excreted as ammonia. So remember, normal serum uric acid level in a male should be 3 to 7 mg per deciliter, and in female it must be 25 mg per deciliter. Total uric acid excreted every day, 24 hours, like you excrete almost 400 to 600 mg. How does this excreted? This is excreted as ammonia. This is excreted as ammonia, and that is what you will be reading. I I don't know how many of you read your amino acid metabolism. In amino acid metabolism, you have your urea cycle, you have ammonia cycle, where you will be reading it. Here, I'm not going to discuss in detail about that. Remember that uric acid, 400 to 600 mg per 24 hours, it is going to be excreted. So what happens? There is overactivity of. So whenever, can you see it? So you have this xanthine oxidase enzyme. When there is going to be a overactivity of xanthine oxidase enzyme, whenever there's overactivity of xanthine oxidase enzyme, what happens is, so xanthine oxidase is a rate limiting step here. So your xanthine oxidase is a rate limiting step here. So whenever there's a overactivity of your xanthine oxidase, it increases the uric acid level. Okay, it increases uric acid level in the blood. Okay, so especially what happens is, if you are going to go into night time, okay, so in the increased uric acid level on the blood la irukum. So it gets projected out abdina when you're going to have a well fed. So when you're going to take a good protein rich food. Similarly, when at night time la usually your pH will be more. Usually your pH will be more at night time. So acidic pH irukumbodhu uric acid level nariya irukum. Similarly, alcoholism. So alcoholic patients ku vandu pathina lactic acid will be more in the urine. So lactic acid will be more in the urine. So this is the uric acid level increased. Okay, so at one point of time, what happens is, all this uric acid gets precipitated. It gets precipitated. Okay, enga precipitate agun pathina at the cold areas. At cold areas, I will tell. So cold areas na enga, your knee, okay, in your joints, in your first toe, in your kidney. Okay, ange ella edathilime it is going to get it is going to sit down. Okay, it is going to go and sit down. Okay, uric acid, either it can go as a monosodium urate crystal. Monosodium urate crystal. It forms monosodium urate crystal. Okay, and this is going to sit in the kidney. It is going to sit in the kidney. It is going to sit in the first toe. It is going to go and sit in the MCP joint. It is going and sit in the MCP joint. All your small joints. It is going and sit in all your small joints. And when they go and sit on all the small joints, the disease, you call it as gout. Now you understood? இவ்வளோ பெரிய ப்ராசஸை நம்ம எதுக்கு படிச்சோம்? இந்த சிம்பிள் திங், இந்த கவுட் அப்படிங்கிற ஒரு ருமட்டாலஜியில உங்களுக்கு ஃபைனல் இயர்ல மெடிசின்ல நீங்க படிக்க போற அந்த ஒன் பர்டிகுலர் இம்பார்ட்டன்ட் டாபிக், ஒன் பர்டிகுலர் இம்பார்ட்டன்ட் எஸ்ஏ, இம்பார்ட்டன்ட் சப் கிளினிக்கல் டாபிக் விச் யூ ஆர் கோயிங் டு ரீட், அதோட பேஸ் எங்க வருதுன்னா உங்களோட பயோகெமிஸ்ட்ரியில வருது. ஒன்ஸ் யூ அண்டர்ஸ்டாண்ட் திஸ், தட் இஸ் கோயிங் டு பிகம் ஈஸி ஃபார் யூ ஆல். ஸோ யூ ஆர் கோயிங் டு ஹேவ் அ கவுட். ஓகேவா? இந்த கவுட்ல என்ன ஆகும்னா, தேர் இஸ் கோயிங் டு பி அ yellowish, or I can call it as greenish crystals are going to be there. So this is a negatively bifringent. They are negatively bifringent. So all these points they will be asking in your Viva, guys. Kindly note it down. Negatively bifringent and negatively bifringent are. So this is the structure. Okay, wow. So this is what you all need to remember. Okay, now I have a gout. Okay, now I am seeing a patient with a gout. Now let's move into a clinical biochemistry. So you are going to see a patient with a gout. Someone put in the chat box, what will be your treatment for the gout? If you understand the pathology of your gout, okay, what is going to be a treatment of your gout? Can someone tell me in the chat box, what is going to be your gout? I'll give you time. Kindly put in the chat box, what is going to be your treatment of choice in the gout? Come on, guys. What is going to be the treatment of choice in the gout? I want you to respond. It will be very interesting when you respond, when you're going to interact with me, so that I can take the session very interestingly. Okay, so I think so no one is interested to respond back. Okay, no problem. So the answer is going to be xanthine oxidase inhibitor. So what you will do is, basically you will be inhibiting your xanthine oxidase. So overactivity of xanthine oxidase is the one that causes gout, isn't it? So what I'm going to do is, I'm going to inhibit your xanthine oxidase enzyme. So when I'm going to inhibit the xanthine oxidase enzyme, what happens is, okay, when I'm inhibiting the xanthine oxidase enzyme, there will be less uric acid formation. When there is less uric acid formation, what happens? Uric acid synthesis, less uric acid breakdown. So you can go, okay, you can either inhibit your xanthine oxidase, or what you can do is you can inhibit your purine production itself. You can inhibit your purine production. Purine production inhibit pannalam. Okay, xanthine oxidase inhibitor for example enna allopurinol. This is the one you are going to read next year in your pharmacology. Inge neenga padichiteenga in the simple concept enjina inge nama padichikalam. So xanthine oxidase inhibitor you are going to give for gout. That is going to be a allopurinol. Similarly, you want to inhibit the purine production. So how you are going to inhibit the purine production? You need to inhibit the rate limiting step of your purine production. Purine production like a rate limiting step which you are going to inhibit. Okay, which you are going to inhibit is, can someone? Can someone? Okay, I'm just leaving it to you. Just respond. Now, other one is going to be you have to increase the uric acid breakdown. You have to increase the uric acid breakdown. How do you increase the uric acid breakdown? By giving a drug called as rasbunicase. Okay, rasbunicase is a drug which increases the uric acid breakdown. Allopurinol, it inhibits your xanthine oxidase, and you can give you can inhibit the purine production. How do you inhibit the purine production? Now you know what is the steps of your purine production, isn't it? This step you know, right? So what happens is we can inhibit at certain stages. For example, what I can do is I can inhibit at this particular area. If I inhibit the formation of your IMP, and if I'm going to inhibit the formation of your IMP, I can inhibit the formation, or what I can do is I can inhibit this particular stage. If I'm going to inhibit the IMP converting into GTP, when I want to convert the IMP conversion into GTP, I'm going to inhibit. So that is going to be your IMP dehydrogenase inhibitor. IMP dehydrogenase inhibitor. What is the example of IMP dehydrogenase inhibitor? 6-mercaptopurine, 6-mercaptopurine, and you have azothioprine. These are the two drugs that are going to inhibit your IMP dehydrogenase. So this is the clinical biochemistry which you are have to read it now itself. Okay, so the pharmacology, why you wanted to go and learn next year? So IMP dehydrogenase inhibitor, you can inhibit the production of purines by 6-mercaptopurine and azothioprine. Okay, next one is going to be you can inhibit the production of your purine by inhibiting this PRPP aminotransferase enzyme. So in the PRPP aminotransferase enzyme, inhibit panla. How do you inhibit this PRPP aminotransferase enzyme? But by giving a drug called as ribavirin, and you have this mycophenolate mofetil. Okay, so these are the two drugs which inhibits your PRPP aminotransferase, glutamyl aminotransferase. When you inhibit this PRPP glutamyl aminotransferase, they are going to be helpful for you for decreasing the production of your purines. Now you understood, right? So you understood, right? Now we have actually discussed two important steps for decreasing the purine production. Increasing the uric acid breakdown, we discussed rasbunicase. And what is the inhibitor of your xanthine oxidase? We have allopurinol. So we discussed three important things. Now tell me, now I will discuss a very important part again. The next important part is going to be, we already discussed here. Can you see, or can you just go back to this important concept? So guanine gets converted into GMP. Your guanine gets converted into GMP in the presence of SGPRTase. What is this SGPRTase? Hypoxanthine guanine phosphoribosyl transferase. When this hypoxanthine guanine phosphoribosyl transferase is not working, unfortunately, you already know that brain is a salvage pathway. So brain la salvage pathway, and the salvage pathway la in the SGPRTase enzyme inhibited abdina. What happens? Your brain cannot synthesize DNA. If this enzyme is not working. Okay, what happens? Your brain cannot synthesize DNA. If there is no synthesis of DNA, it leads to mental retardation. And self-mutilation. What is this disease called as? This disease is called as Lesch-Nyhan's disease, Lesch-Nyhan's syndrome. I hope that your book will be having this, but you will not have the idea how this disease came, isn't it? So your Lesch-Nyhan syndrome, because of the not working of SGPRTase, hypoxanthine guanine phosphoribosyl transferase, when this enzyme is not working properly, they are going to give rise to a mental retardation that is called as your Lesch-Nyhan syndrome. Is it clear, guys? Lesch-Nyhan syndrome, how it occurs? Total absence of your SGPRTase. ஒருவேளை partial absent ஆகுது. So instead of completely not working, some are not working. Okay, some are working னு வச்சுப்போம். ஓகேவா, ஒரு 10 பேர்ல 2-3 பேர் கிட்ட ஒர்க் பண்றாங்க, மீதி யாருமே ஒர்க் பண்ணல அப்படின்னா there will be a partial mental retardation, and that is called as your Kelly-Seig-Miller disease, Kelly-Seig-Miller syndrome. Okay, so Lesch-Nyhan syndrome is nothing but when this SGPRTase enzyme is not working, you call it as a Lesch-Nyhan syndrome. When your SGPRTase enzyme is partially working, you call it as a Kelly-Seig-Miller syndrome. Kelly-Seig-Miller syndrome. So what is this Kelly-Seig-Miller syndrome? It is going to be a partial absence of your SGPRTase enzyme. Okay, is it clear? You understood two things. Now the third important thing I wanted to tell you is this severe combined immunodeficiency, SIDS, severe combined immunodeficiency. This is one of the disease. So இங்க என்ன இருக்குதுன்னா, நீங்க guanine பார்க்குற அந்த enzyme problem இருந்தது இல்ல, அப்ப you have this mental retardation, you have this two disease, that is your Kelly-Seig-Miller syndrome and your Lesch-Nyhan syndrome. Similarly, when there is going to be a problem in the production of AMP from your adenosine, you have this AMP production. Okay, adenosine landi AMP production. Okay, adukku adenosine phosphoribosyl transferase enzyme irukilla, and the enzyme la edavadhu problem irundaduna, and the ribonucleotide reductase enzyme la edavadhu problem irundaduna, again DNA synthesis nadakadu. Ena brain la purines ellame both your adenine and guanine are going to come in that particular salvage pathway. So guanine production illana, guanine landi GMP varlana, you told these diseases at the beginning. Now what I am telling is, when adenine cannot get converted into adenosine monophosphate in your brain, that leads to your SCID, that is your severe combined immunodeficiency. It can lead to tuberculosis. Okay, tuberculosis, it leads to tuberculosis, TB lymphadenitis. Okay, so these two important diseases comes because of the deficiency of your adenosine ribosyl transferase in your enzyme deficiency or enzyme not working properly. Now you understood this, right? So idadana enoda PDF la I would have given you detailed explanation. Gout is an inherited disorder of purine metabolism characterized by hyperuricemia. You have a primary, secondary. So the enzyme which is going to be a problem is PRPP synthetase, SGPRTase. So all these things. Now I told you how secondary gout occurs. What are the clinical features? Clinical features la neenga now vechukka vendiyadhu, metatarsophalangeal joints la you will be having a uric acid deposition, that is called as tophi, and you have synovial fluid la you have monosodium urate crystals. These monosodium urate crystals can be demonstrated like this. So when it deposits in the renal medulla, you have this renal caliculi or renal damage can occur. Can you see it? This is how your gout will be there. Okay, what will be your treatment? I told you already, allopurinol, as Krishpa Sharma told. Very good. Allopurinol is a competitive inhibitor of xanthine oxidase, and that will be helping you for reducing the urate production. And you can increase the renal excretion by uricosuric drugs, and you have to take the patient, decrease alcohol intake or decrease purine intake, and anti-inflammatory drug like aspirin can be given and colchicine can be given. And I told you about the Lesch-Nyman syndrome. This is your Lesch-Nyman syndrome. A spotter can be kept in your exam. This is Lesch-Nyman syndrome. I hope you can see the screen. And you have this hypouricemia. What is the other condition? When the uric acid is not present. That is what I told you. Adenosine deaminase deficiency can cause immunodeficiency, severe combined immunodeficiency. Okay, and you can also have your that's all. So you have your adenosine deaminase deficiency, severe combined immunodeficiency. T cell, B cells are going to be dysfunctional. Accumulation of inhibit the ribonucleotide reductase enzyme. Hope so you understood this. Now, can someone tell me, shall we continue with the topic or shall we close it and shall we continue it tomorrow? Can someone put in the chat box? If you want me, I will continue, or else we will continue in the next class. What I will do is Pyramidine. I will put it as a video again. I don't want to take live class in Pyramidine. Pyramidine, we will put it as a live class. We will be putting it as a video, and then I will continue with the DNA, RNA, and we can go with the replication, transcription, translation from tomorrow evening. Is it clear, guys? If you have any doubt, you can put in the chat box. I think 1 hour, exact 1 hour, we are just going to complete. The first topic, a base of this, purine pyrimidines are important. They are going to be forming the base of your DNA, RNA formation or DNA, RNA production. That is why I have taken so much of time to make you understand. And basically, this is a place where you get fear, because seeing all those structures in your exam, what happens is you will get a fear out of your own without knowing itself. So that is why we have made sure that the basics are very clear. So that what we will do is we will build up the concepts later, and we will talk about the pyramidine tomorrow. I'll just put it as a video. You can come after your college, you can come and you can see that video. And after completing it around 7 o'clock or around 8 o'clock, what we will do, we will start with the DNA. So clear? Thank you so much, guys. Thank you so much. So we